God, a murder has been committed! The sword was left at the crime scene with the victim's blood on it. The DNA from the crime scene must be replicated in order to have enough for gel electrophoresis testing. In order to understand the process of PCR, let's take a quick look at the DNA molecule. DNA is a double helix molecule containing nucleotides. Each nucleotide is made of a nitrogen-based phosphate group and a sugar. A swab of the blood was taken for further testing. The swab with the DNA is placed in a solution of cell lysis buffer. This buffer causes the cells to break apart, thereby releasing their DNA into the solution. The DNA is now ready for the PCR process. In PCR, polymerase chain reaction, you place samples of DNA along with polymerase, primers, nucleotides, buffer, and mineral oil into small test tubes and then into the PCR machine. PCR has 3 steps. In the first step, the temperature is set to 90 degrees Celsius or 96 degrees Celsius. The DNA is then denatured or separated into 2 strands. In step 2, the temperature is lowered to 50 to 68 degrees Celsius in order for the primers to anneal or attach. In step 3, the sample is heated again to 72 degrees Celsius in order for the polymerase to replicate or extend the DNA from the primer locations. This is also known as extension. This process is repeated until a usable amount of DNA is created about 30 times. Now that we have enough DNA, we will use the gel electrophoresis process to compare the DNA samples from the crime scene with our suspects. A restriction enzyme is added to the samples. The enzyme cuts the DNA into smaller fragments at specific points such as between the A and T bond. The cuts will produce different sized pieces of DNA in different samples. If 2 DNA samples are the same, the resulting pieces will be the same sizes. The different DNA samples are now loaded into the gel. The lid is attached and the power is turned on, producing an electric current in the buffer that surrounds the gel. The different sized pieces of DNA move through the gel at different rates, giving us a nice spread in the gel. When a nice spread is achieved, the power is turned off and the lid is removed. The sample is usually stained and photographed. If there is a match between the suspect's DNA and the blood on the sword, then the lawyers will have valuable evidence in order to convict the guilty party.